Welcome back. We are about to tackle some hard work. You know what? Knowing the content and all the definitions and being able to recognize graphs and say whether they are geometric or logistic, that's only part of life sciences. There's a whole other part that sometimes you forget to do, and that is learning the skills. And unless you practice the skills, you'll find that you can answer the multiple choice questions, you can answer the terminology, but when it comes down to producing graphs, for example, writing paragraphs, suddenly your skills let you down. So in this part of our lesson, we're going to practice, and this is a typical assessment task where you are in fact going to look at a set of data and produce a graph. So I hope you have got your pen and paper, and I hope you're ready to draw something absolutely amazing. Let's see where we're going now with logistic growth curves. So I'm going to set up an experimental situation for you. We're going to use a population of yeast. Now, yeast are fungi. They belong to the kingdom fungi. And yeast, although you're familiar with mushrooms and larger fungal species, yeast are in fact microscopic unicellular little fungi. They are asexual reproducers, and here in this photograph you can see that a mature yeast cell starts to make little buds, and eventually these buds grow bigger and bigger, and they break off the original cell and they can start growing as independent organisms themselves. But now here's something interesting. One yeast cell, here's a little bud breaking off, can produce multiple buds at the same time. So yeast cells, their population grows very quickly. So our practice example that we're going to be looking at focuses on a population of yeast cells. Now, here's our experiment, and my first instruction to you whenever you get one of these types of questions is to read. Read it carefully. Use highlighters or underline things that you think are going to be important. So in an experiment, yeast cells were grown in a glucose solution. So that's the food. That's the nutrient source. And they were grown in a test tube. All right? Now, we must remember that a test tube has limited size. It's kept at a temperature of about 30 degrees, and every 2 hours a little drop was taken from our sample and it was placed onto a microscope slide, and we counted the number of cells in a particular unit area, and we are going to accumulate these results in a table, in a data table. So I think you understand the setup. We're growing a population. Our population is going to receive food. It is going to be kept at an ideal temperature, but it's in a limited amount of space. And we know that these yeast are going to be reproducing quite rapidly, and we're going to take a sample every 2 hours, and we're going to have a little grid on our microscope slide, and we're going to count the population of yeast cells. All right, we're going to produce this kind of data table. Now, once again, before you move on to the questions and find out what it is you have to do, make the information and the data your own. Make sure that you understand the context of this question before you shoot ahead into the questions and you've missed something. So in this question, we're provided with the data table. Let's look at it carefully so that we know precisely what it is we're looking at. One of our variables is time, and this we should expect because we were told that every 2 hours a sample is taken from the population. So we must expect at time 0, 2 hours later, another 2 hours later, and our investigation went on for 18 hours. What information we were measuring was counting those little yeast cells. We're actually sitting going 1, 2, 3, 4, we're counting those cells down the microscope. And at the beginning, time 0, we find in our unit area, which is going to be remaining constant throughout our investigation, we see we had 10 yeast cells. We come back after 2 hours and we take another sample. Same unit area, same size, that is a controlled variable. Now we see that our size of our population is in that same space, 30 yeast cells. 2 hours later, it's gone up to 70, to 175, and we can see what we call a trend. And a trend, another name for a trend in life sciences, is a pattern. We can see a pattern developing. As time goes on or increases, so our numbers of the yeast cells are increasing as well. Now, that's all very well and good. We've got an overall trend that we can describe just from our data table. But is it a completely accurate description or an accurate picture of what is happening to this population inside our test tube? If we took this information from a data table and we plotted it on a graph, we would be able to understand the trend a lot more accurately. Instead of saying, yes, as the one variable increases, so does our other variable increase, we'd be able to say, but hold on, there's something more important going on. It's not just a simple, this is increasing and that is increasing at the same time. There's something more complicated going on in terms of our population growth. And that is where drawing a graph is so important. So it's little surprise that our first question in this activity asks us to draw a graph. Showing, what are we showing in our graph? The population growth curve that resulted from this investigation. Now, before you start, you bring to the exam question or the test question all your knowledge about growth curves. We know that the growth curve could be a J-shaped curve, or we're going to see possibly an S-shaped or a sigmoid-shaped growth curve. We know that if it's a J-shaped, we're looking at geometric. If we see an S-shape, we're looking at logistic growth curves. So we're bringing all this knowledge that you've studied so hard to the practical situation. We're asked now to draw the graph. So this is where your pen and paper becomes very important, because I'm not just going to do this job, you've got to do it as well. All right, so as we work our way through this example, you're working just as hard as I'm working. Our first step is to set up a set of axes. Now, you know that this is a diagram. A graph is a kind of diagram. Please don't, if you're doing this in a test or an exam situation, try and squash it in to the bottom of the page because, look, I've got 5 lines. I can draw my graph here. Please make sure that you draw the graph nice and big. Maybe go over onto a brand new page so that you've got lots of space for that graph. So we're going to draw our axes. What kind of graph are we actually going to draw? Is it going to be a line graph? Is it going to be a bar graph, a histogram? I think you understand from what we've been talking about growth curves is that we're looking at continuous variables, which means if our data is continuous, we're drawing line graphs. So we've got our axes set up. We've just drawn the axes. Have you done that? I'm checking. Yes? Drawn it? Good. Let's move on. Next thing, we have to make sure that we plot our variables from our data table into this graph. We're going to go, time is very easy. Time is our independent variable that we're controlling. We measured the population every 2 hours, so that's easy to plot. We now look at our smallest number of yeast cells and our largest number of yeast cells, and we must make sure that our y-axis is going to accommodate going from 10 all the way up to 660. Right, so we've got an idea of the scale, and we have labeled our axes as well. Now we have to plot our points. Are you ready to plot the points? Let's go. At time 0, there were 10 yeast cells. So time 0, we've got 10 yeast cells. Plot a little pencil mark. At 2 hours, we were up at 30 yeast cells. There's our little mark. At 4 hours, we were up at 70. And so on. We're going to plot each individual point. And the next logical step is now to draw a smooth line joining the points. What kind of graph and trend do you now see emerging? It's not as simple as time increases, so do number of yeast cells increase. Something else is happening here. We're seeing a lag phase, an acceleration phase, a deceleration phase, and we're seeing some kind of stabilization or equilibrium happening towards the end of our time in terms of this investigation. So if we look at the graph, we have got a sigmoid graph. Let's now have our final point to drawing the graph. It must have a heading, and the heading must reflect both variables, time as well as the growth of the population. On your graph, indicate the lag phase, the exponential phase, and the stable phase. Here's our lag phase. Here is our exponential phase, and we're going into a stable phase up at the top. Describe the shape of the graph and indicate what kind of graph this is. Well, we're way ahead of this question. We said that it was an S-shaped graph, or the word that you could use is sigmoid, and that is characteristic then of logistic growth. All right, got it? Let's move on. During which time period was there the greatest increase? Now, the greatest increase is going to refer to the gradient or the slope of our line. So let's see, where was our slope of the line the steepest? And we're looking at between 6 hours and 8 hours. After 6 hours, there was 175 yeast cells. There we go, 175. How long does it take to double that population? Well, if we look that in just 2 hours, we're up at 350, we have doubled the population in simply 2 hours. Why do you think the growth rate slowed down? Well, here we've got a lot of factors. So you'd be able to write a couple of paragraphs or a sentence or two, and you'd be able to say things like increase in the amount of wastes produced by the yeast, competition for nutrients, overcrowding. All right, you can talk about all the limiting factors that slow down population growth. I've put together a little summary for you to cover what we've done in this particular lesson, and I hope that you're able now to be able to talk about growth curves with a lot more confidence and relate your idea of growth curves to how populations change over time. But that's enough information for today. So from me, Mama H, to you out there working very hard, I'll see you in our next lesson. But for today, goodbye.